Hello, welcome to Biology, the scientific study of life. Gene expression can be regulated at any stage, but the key step is transcription. All organisms must regulate which genes are expressed at any given time. Both unicellular organisms and the cells of multicellular organisms must continually turn genes on and off in response to signals from their external and internal environments. The cells of a multicellular organism must also regulate their gene expression on a more long-term basis during development of a multicellular organism. Its cells undergo a process of specialization in form and function called cell differentiation, resulting in several of many differentiated cell types. The mature human body, for instance, is composed of about 200 different cell types. Examples are muscle cells and nerve cells. Differential gene expression. A typical human cell probably expresses about 20% of its genes at any given time. Highly differentiated cells such as muscle cells express an even smaller fraction of their genes. Although almost all the cells in an organism contain an identical genome, the subset of genes expressed in the cells of each type is unique, allowing these cells to carry out their specific function. The differences between cell types, therefore, are due not to different genes being present, but to differential gene expression, the expression of different genes by cells with the same genome. The genomes of eukaryotes may contain tens of thousands of genes, but for quite a few species, only a small amount of the DNA, about 1.5% in humans, codes for protein. Of the remaining DNA, a very small fraction consists of genes for the RNA products such as ribosomal RNA and transfer RNA. Most of the rest of the DNA seems to be non-coding, although recently researchers have learned that a significant amount of it may be transcribed into RNAs of unknown function. In any case, the transcription proteins of a cell must locate the right genes at the right time, a task on a par with finding a needle in a haystack. When gene expression goes serious imbalances and diseases, including cancer, can arise. Summarizes the entire process of gene expression is in a eukaryotic cell, highlighting key stages in the expression of a protein coding gene. Each stage depicted in this figure, notice that, is a potential control point at which gene expression can be turned on or off, accelerated or slowed down. Only 40 years ago, an understanding of the mechanisms that control gene expression and in eukaryotes seemed almost hopelessly out of reach. Since then, new research methods, including advances in DNA technology, have empowered molecular biologists to uncover many of the details of eukaryotic gene regulation in all organisms. The expression of specific genes is most commonly regulated at transcription, often in response to signals coming from outside the cell. For the reason, the term gene expression is often equated with the transcription for both prokaryotes and eukaryotes. However, the greater complexity of eukaryotic cell structure and function provides opportunities for regulating gene expression at additional stages. In the following three sections, we will examine some of the important control points of eukaryotic gene expression more closely. Notice that this figure stages in gene expression that can be regulated in eukaryotic cells. In this diagram, the colored boxes here indicate the processes must often regulated. The nuclear envelope separating transcription from translation in eukaryotic cells offers an opportunity for transcriptional control in the form of RNA processing that is absent in prokaryotes. In addition, eukaryotes have a greater variety of control mechanisms operating before transcription and after translation. The expression of any given gene, however, does not necessarily evolve every station. For example, not every polypeptide is cleaved. As in prokaryotes, transcription initiation is the most important control point. Notice that this signal and represent this one chromatin, and this is nucleus. What's happened inside the nucleus? Chromatin. Notice that gene gene available for transcription. Before this one, chromatin modification, DNA unpacking involving histone acetylation and DNA demethylation. Notice that this one's transcription, RNA, exon, intron, primary transcript, and this one RNA processing, tail and cap. Notice that mRNA cytoplasm transport to cytoplasm. Notice these are cytoplasm, yellow one. Degradation of mRNA. Notice that translation, polypeptide, these are polypeptide, and cleavage chemical modification, transport to cellular destination, and this one active protein, degradation of a protein and degraded of protein. Regulation of a chromatin structure. As mentioned earlier, the structure organization of a chromatin not only packages of cells DNA into a compact from that fits inside the nucleus, but also DNA into a compact from that fits inside the nucleus, but also it is important in helping regulate gene expression. Genes within heterochromatin, which is highly condensed, are usually not expressed. The repressive effect of heterochromatin has been seen in experiments in which a transcriptional active gene was inserted into a region of heterochromatin in yeast cells. The inserted gene was no longer expressed. In addition, the location of genes promoter relative to nucleosomes and to the sites where the DNA attaches to the chromosome scaffold or nuclear lamina can also affect whether it is transcribed. A flurry of recent research indicates that certain chemical modifications to the histones and DNA of chromatin influence both chromatin structure and gene expression. Here we examine the effects of these modifications, which are catalyzed by specific enzymes. Histone modifications. There is mounting evidence that chemical modification to histones play a direct role in the regulation of gene transcription. The N-terminus of each histone molecule in a nucleosome protrudes outward from the nucleosome. Not as shown here. Notice that. Closer look. These histone tails are accessible to various modifying enzymes which catalyze the addition or removal of specific chemical groups. In histone acetylation, acetyl groups CO, CH3 are attached to positively charged lysines in histone tails. Distillation is the removal of acetyl groups when the histone tails of a nucleosome are acetylated. Their positive charges and neutralize it, and they no longer bind to neighboring nucleosomes. Recall that such binding promotes the folding of a chromatin into a more compact structure. When this binding does not occur, chromatin has a looser structure. As a result, transcription protein have easier access to genes in an acetylated region. Researchers have shown that some enzymes that acetylate or decetyl histones are closely associated with or even components of the transcription factors that bind to promoters. In other words, histone acetylation enzymes may promote the initiation of transcription not only by modifying chromatin structure but also by binding to and thus recruiting components of the transcription machinery. Several other chemical groups can be reversibly attached to amino acids in histone tails. For example, the addition of methyl group CH4, notice that, to histone tail methylation can lead to condensation of the chromatin. The recent discovery that this and many other modifications to histone tail can affect chromatin structure and gene expression has led to the histone code hypothesis. According to this model, specific combination of modification rather than the overall level of histone acetylation help determine the chromatin configuration within which in turn influences transcription. Okay, back to a simple model of histone tails and the effect of histone acetylation. In addition to acetylation, histone can undergo several other types of modification that also help determine the chromatin configuration in a region. Notice that yellow represents amino acids available for chemical modification, and this tail one, histone tail, notice the closer look, notice that. DNA, this DNA double helix, and histone tails protrude outward from a nucleosome. In this end view of nucleosome, each type of histone is shown in a different color. The amino acids, the N-terminus tails, and accessible of chemical modification. Notice B also unacetylated histone and this acetylated histone. Notice differences between unacetylated and this one acetylated histone. Acetylation of histone tails promotes loose chromatin structure that permits transcription, a region of chromatin in which nucleosomes are unacetylated forms a compact structure in which the DNA is not transcribed. When nucleosomes are highly acetylated, right, the chromatin becomes less compact and DNA accessible for transcription. Okay, thanks.